Thank you. I'm going to talk about genetic basis for unilateral renal agenesis and heterogeneous stock rats. URA has been described as the urinary tract malformation, and it's also coupled with some reproductive tract malformations. So it's determined as a congenital absence of one kidney. Previously, it has been reported a locus associated with this specific phenotype. It has been named as renal1. It only harbors the KIT gene, so this gene has been described as the most probable candidate gene for this specific phenotype. It has been also reported an insertion located in the intron 1 of this gene. It's an endogenous retrovirus. You have the representation here. It's composed by 3 different elements, 2 different long-terminal repeats, and 1 endogenous retrovirus sequence. This gene encodes KIT proton cogene receptor tyrosine kinase, which is involved in regulation of cell survival and proliferation. Introduction. Previously, Dr. Garrett was able to develop a strain. This is named the HSRA. This strain has been developed from the same genetic background as the one that we are using here, the HS population. This is important because it means that we can assume that the same QTLs responsible for URA are present in both populations. This strain was embedded for unilateral renal agenesis, and it shows an incidence between 50 and 75% of the offspring showing unilateral renal agenesis. This shows that through breeding, it's possible to increase the frequency of those elements that are responsible for URA and increase the incidence of this specific phenotype. The incidence in the present population is around 1.9% of the population. I'm going to show the ACI incidence. This is one of the 8 founded strains of the HS population. Is the one with the highest incidence. It goes between 5 and 15% of the offspring. Additionally, it has been described an incomplete penetrance for this phenotype. The objectives of this analysis were to identify genomic regions associated with URA, controlling for case control imbalance, and to recognize potential loci able to modify the penetrance for this phenotype. Materials and methods, we used 5,600 individuals from the HS population. Those individuals were genotyped for 3.5 million markers, and the case control ratio that we identified was around 1 case per 51 controls. So it's not very extreme, this case control imbalance. You are already familiar with the HS population. I'm only going to talk again about the ACI strain. We used for the association test the R package and SAGE-GDS, applying an additive model for the association. This package is able to control case control ratio when it's extremely imbalanced, and it applies a generalized mixed model association. Here you have a comparison using the standard linear mixed model and SAGE. As you can see here, the former is unable to control type 1 error. You can see an increased rate. On the other hand, SAGE is able to control this error, and it's able to pinpoint highly significant QTLs associated with this type of phenotypes. This representation is showing a thyroid cancer. It's a phenotype with a very skewed imbalance, 1 case in 1,134 controls. Results, I'm showing here the most significantly associated region. It's located in chromosome 14 between the megabase 32 and 37. You have a detail here. We have again the KIT gene right in the middle. So this result is in agreement with previous reports showing that this gene as the most probable candidate gene. Here you have the most significant SNP. We had a question here since we have a total of 1,200 markers above of the association threshold. So we wanted to know if maybe we have multiple QTLs associated with URA. So we run a secondary association analysis fitting the most significant marker as a covariable in the model. And we were able to determine that the most probable result is just one QTL. This SNP was able to account for all the variability present in this specific locus. We perform a secondary analysis using haplotypic probability estimation. We focus this analysis on the associated region, chromosome 14, 32, 37 megabase, and we also focus the analysis on the haplotypic probability for the ACI strain. The goal of this analysis was to determine if maybe this specific region is more frequently inherited from ACI when we are talking about cases. So here you have a representation. This is haplotypic probability by strain. It's showing all 8 strains. I'm showing the subtraction between cases and controls. So if you have a positive value, it means that this probability is more, it's higher for cases. As you can see here, we have the ACA strain. It's more highly probable for cases inside this specific region. The second plot is showing exclusive alleles by a strain. I'm showing here a subset of SNPs that are ACI a subset of alleles that are ACI exclusive. Another subset is not ACI exclusive. It's present in ACI, VOF, and N520. However, when we couple this information with the prediction of haplotypic mosaic for the ACI, we are able to determine that even when those alleles are not ACI exclusive, they are more frequently inherited by this specific strain for cases. So we are able to conclude that for this specific region, the most significantly associated region, there is a higher influence of the ACI strain when we are talking about cases. As I said previously, incomplete penetrance has been also described for this phenotype. If we take a look at the distribution, phenotypic distribution for the genotypes for the most significant marker, we are able to determine that not all the cases are at least carriers for the risk allele, being the allele T. So it agrees with an incomplete penetrance for this phenotype. For this analysis, we performed a similarity analysis. The goal of this analysis is to tackle that issue of an incomplete penetrance. We used the Plum database. We fitted the Jacquard index, and we used as a reference the HSRA strain, and everything was coded in Java. So this methodology is going to tell you how similar are two sequences. So here you have a method example, one reference, the HSRA strain, and 2 different individuals. So you are going to perform a pairwise comparison between each individual against the reference. And the result, similarity score, it's coming from how many elements are in common between the 2 sequences that you are doing the comparison. We used the most probable polymorphisms associated with this phenotype, the insertion of the endogenous retrovirus sequence. We did this analysis in 2 different steps. We did a prediction for transcription factor binding sites in all 3 sequences using transcription factors that are expressed in kidney or kidney-related tissues such as collective tubes. For example, here we have the N01 was discovered as one of those transcription factors able to interact with DNA sequences for those insertions. The second step was to perform the similarity score analysis. So we know that this specific transcription factor is being encoded from this specific locus, chromosome 5, between those locations. We pull out all the SNPs that are inside this locus, and we perform the similarity score analysis. So you are going to end up with a comparison for the N01 between each individual and the N01 from the HSRA, the reference sequence. Here you have the results for this analysis. A number of transcription factors were identified as significantly different when you group the individuals based on the genotype for the most significant marker. They have been color-coded The green represents the highest similarity with the HSRA, and the yellow the lowest similarity. If we take a look, for example, to this transcription factor, the HNF1A, individuals that are grouped as TT for the most significant marker, they are highly similar to the HSRA strain, meaning that this transcription factor had been selected for in those specific individuals. This transcription factor encodes a gene regulator of the proximal tubule, and it's involved in apoptosis. Those processes are required for organogenesis of kidney. As a result, we were able to identify some transcription factors that have been selected for in the HSRA strain. The mechanisms that we believe it's related in this phenotype had been previously described in stimulating cells. Liu identified that higher expression of the KIT ligand, it can be determined as KIT activation, can drive proliferation. On the other hand, lower expression of the KIT ligand, it can drive differentiation, cellular differentiation. Those two processes are key involved in kidney organogenesis, since proliferation is required for hereditary BAT invasion and differentiation is required for metanophytic rudiment differentiation. All those, a balance between those two processes are required for a normal branching morphogenesis, which is required for organogenesis of kidney. So this balance between proliferation and differentiation are key. We believe that key expression can drive differences between those two processes, and some of the transcription factors that we identified as being selected in the HSRA can modulate that expression, having an effect on this specific equilibrium. And that could be driving that specific phenotype that we identified, the URA phenotype. Conclusions, we identify a region associated with URA, controlling for case controlling balance, and we were able to identify some potential loci able to modify penetrance for URA. And further research is needed. Thank you.